To all my longtime viewers, you already know that Fact Cove has never been about perfectly matching every visual, it's always been about delivering accurate, fascinating facts. And if you're new here, welcome. I want you to know the same thing from the start. Fact Cove isn't about matching every visual, it's about giving you the facts that matter. On the island of Guam, the forests fell silent. A place that was once a symphony of birdsong became an eerie, quiet jungle. The cause wasn't a mystery. It was an invasion. An invasion of brown tree snakes, which accidentally arrived sometime in the late 1940s or early 1950s, likely as stowaways in military cargo. With no natural predators on the island, their population exploded to terrifying densities, in some areas, reaching an estimated 13,000 to 20,000 snakes per square mile. They devoured the island's wildlife, helping drive 9 of Guam's 12 native forest bird species to local extinction. They climbed power poles, causing hundreds of electrical outages every year. They became one of the most infamous, most studied invasive species on the planet. But behind this story of ecological devastation, a deeper scientific mystery was brewing. One that would challenge everything we thought we knew about survival, inbreeding, and the very code of life itself. The entire snake population on Guam was believed to have started from just a handful of individuals. By all scientific logic, this should have been a genetic death sentence. A tiny founding group creates what's called a genetic bottleneck. Inbreeding should have weakened them, making them less able to adapt and more vulnerable to disease. They should have died out. Instead, they conquered. So, what was their secret? For decades, biologists were baffled. The answer was hidden deep inside their DNA, and it would take a new generation of technology to finally find it. The invasion and the paradox. To truly grasp the scale of this paradox, you have to understand what Guam was like before, and what it became. It's an isolated island in the western Pacific, a lush, tropical environment. For millennia, its native birds evolved without snake predators. They were naive, nesting in the open, with no defenses against a creature like the brown tree snake. When the snakes arrived, it was a massacre. Species like the Guam rail and the Micronesian kingfisher vanished from the wild, now surviving only in captive breeding programs. The loss of these birds wasn't just a tragedy for wildlife, it triggered a cascade of ecological consequences. Without birds to pollinate flowers and disperse seeds, the very structure of the forest began to change. The ecosystem started to unravel. And the snakes were everywhere, in backyards, in houses, and famously, wrapped around electrical wires, plunging parts of the island into darkness. This overwhelming success flew in the face of a fundamental principle of population genetics, the founder effect. When a new population is established by a very small number of individuals, its gene pool is drastically limited. Imagine a giant bag filled with thousands of marbles of every color. That's a healthy, diverse population. A genetic bottleneck is like reaching in and pulling out just a few marbles. All future generations will only have the colors of those few marbles. This lack of genetic diversity is usually a major disadvantage. It increases the chances of harmful mutations becoming widespread and reduces a population's ability to adapt to new challenges, like diseases or a changing environment. Scientists knew the Guam snakes came from a bottleneck, but the snakes didn't just survive, they thrived in ways that seemed impossible. The paradox was, how? The old science and deepening mystery for years, scientists trying to solve this puzzle used the best tools they had. They studied the snake's DNA using what is now considered standard, or short-read, sequencing. This method is excellent at spotting tiny, single-letter changes in the DNA code, what scientists call SNPs, or single nucleotide polymorphisms. These are like finding individual spelling mistakes in a massive book. When researchers looked for these SNPs in the Guam snake population, they found exactly what they expected, not a lot of diversity. The genetic code from one snake looked remarkably similar to the next. This confirmed the theory of a severe genetic bottleneck. It painted a picture of a genetically impoverished population. But that just deepened the mystery. If their DNA was so uniform on this level, how were they so successful? How did they overcome the new diseases they encountered? How did they become such ruthlessly efficient hunters in a new environment? The data from the old science and the reality on the ground were in direct opposition. It was as if they were reading a book, spotting a few typos, but completely missing the fact that entire chapters had been rearranged. A technological revolution. The breakthrough came from a new way of reading DNA. If the old method was like looking at a book with a magnifying glass to find spelling errors, the new method was like being able to read entire paragraphs, pages, and even chapters all at once. This new technology is called long-read sequencing. A team of researchers from the University at Buffalo and the U.S. Geological Survey decided to apply this powerful new tool to the brown tree snake mystery. Instead of just looking for those single-letter changes, long-read sequencing allows scientists to see massive, wholesale changes to the genome. It can identify sections of DNA that have been deleted, duplicated, inverted, or moved to a completely different part of the chromosome. These are called structural variants. Think of it this way: a single spelling mistake might change a word. But duplicating a whole paragraph or deleting a critical sentence can change the entire story. These structural variants were largely invisible to the older sequencing methods. For decades, we had been looking at the snake's genetic blueprint with one eye closed. The question was, what would we see when we finally opened both? The big reveal, a hidden world of variation. When the results from the long-read sequencing came back, the science team was stunned. A recent study found that the genome of the inbred brown tree snake wasn't a barren desert of uniformity. It was hiding a secret jungle of diversity. In their analysis, the researchers identified more than 19,000 structural variants. 19,000 locations where the DNA was dramatically different, entire sections copied and pasted, deleted, or rearranged. This was the hidden genetic variation that scientists had suspected must exist but could never prove. In fact, one study suggested that these large structural variants affected significantly more of the genome than the simple single-letter changes that had been studied before. The snakes weren't nearly as genetically impoverished as once thought. They were carrying a different, more dramatic, and previously unreadable form of genetic diversity. They had survived the bottleneck not just because they were lucky, but because they were carrying a hidden genetic toolkit. The paradox was beginning to unravel. The snakes that shouldn't have been able to adapt had a wealth of genetic options to draw upon, hidden in plain sight. The snake's secret weapons, smell and immunity. The discovery became even more fascinating when the researchers looked at where these thousands of structural variants were located. They weren't scattered randomly. They appeared to be heavily concentrated in two specific and very important sets of genes, those related to the immune system, and those related to olfaction, or the sense of smell. This finding could be the key that unlocks the entire mystery. For an invasive species, a powerful immune system and a superior sense of smell are nothing short of secret weapons. First, immunity. Arriving on a new island means encountering new pathogens, bacteria and viruses the snake's body has never seen. A population with low diversity in its immune genes would likely be wiped out. But the structural variants may have given the brown tree snakes a diverse and flexible set of immune genes, allowing them to fight off new threats and thrive. Second, and perhaps even more critically, was their sense of smell. Brown tree snakes are nocturnal hunters that rely heavily on chemical cues to find prey. They constantly flick their forked tongues, tasting the air and delivering scent particles to a specialized organ. An enhanced sense of smell, potentially turbocharged by thousands of genetic variations, would have made them extraordinarily effective predators on an island full of unsuspecting prey. This heightened sense of smell might also solve another behavioral puzzle. In their native range, brown tree snakes are known to be cannibals. Yet on Guam, there's little evidence they prey on each other, even at their high population density. One intriguing hypothesis suggests their sophisticated sense of smell may allow them to recognize each other as close relatives, more like siblings than prey, preventing the population from eating itself into decline. The lingering question: the discovery of this hidden diversity answered the how, but it opened up a new and equally intriguing question: the when. Did these thousands of structural variants already exist in the handful of snakes that first arrived on Guam, representing a lucky draw of pre-existing adaptability? Or did some of this incredible variation emerge after the invasion began? Some research suggests that the stress of a severe population bottleneck might actually accelerate the rate at which these large structural variants form. The process of intense inbreeding could, paradoxically, be a crucible that forges new genetic diversity in this large-scale format, even while it erases small-scale diversity. To know for sure, scientists would need to perform the same long-read sequencing on brown tree snakes from their native range in Australia and Papua New Guinea and compare the genomes. If the native snakes also have this high number of structural variants, it would suggest the Guam snakes simply got lucky. But if the native snakes have far fewer, it could point to a radical new idea: that rapid, large-scale evolution can happen in the immediate aftermath of an invasion. For now, it remains an open and exciting question at the frontier of genetics. Broader implications for a planet in flux. The story of the brown tree snake is bigger than just one snake on one island. It has profound implications that ripple across biology. For decades, a primary concern for conservationists working with endangered species has been the loss of genetic diversity in small populations. Species like the cheetah or the black-footed ferret are famous for their genetic bottlenecks. We have often assumed these populations are evolutionary dead ends. But the brown tree snake challenges this assumption. What if, like the snake, some endangered species are also harboring hidden structural variants we simply haven't been able to see? It's possible that some species we've worried are genetically doomed might have more resilience than we realize. This discovery is a wake-up call, urging conservation geneticists to adopt long-read sequencing to get a more complete picture. On the flip side, it's a chilling message for managing invasive species. It suggests that a small number of founding individuals isn't always a guarantee that an invasion will fail. Some species might be genetically preloaded for colonization, carrying a secret arsenal of structural variants that make them incredibly adaptable. The brown tree snake provides a powerful, and terrifying, model for how an invasive species can beat the odds. So, the inbred snake that should have failed didn't just survive, it flourished by rewriting the rules. The mystery of its success wasn't magic, it was a form of genetic diversity that had been hidden from us until now. The silent forests of Guam tell a story of ecological devastation, but the snake's DNA tells a new and surprising story about resilience, adaptation, and the hidden power of the genome. It shows us that even when a population looks uniform on the surface, evolution can be working in powerful and unexpected ways underneath. If you found this dive into the secrets of DNA fascinating, consider subscribing for more stories from the cutting edge of science. And for another look at how animals defy expectations, check out our video on how crows are much smarter than you think. Thanks for watching.